Hello, my name is Dr. Joshua Sebree, and this afternoon I'm going to be talking to you about planetary science and what it means to be a planetary scientist and how planetary scientists go about doing some of their work, both in a laboratory type setting and out in the field. So we'll go ahead and get started here. I do apologize for not being able to do this in person. We're actually in the field right now doing research underground at Wind Cave as you are watching this video. So, sit back and enjoy and learn about some of what planetary science is about, and we will begin. So, who am I? I am a chemist, I am an astrobiologist, I'm a father, I'm a teacher, I'm a professor. Formerly, I work at the University of Northern Iowa. While there, I teach undergraduate students and I work with them on how to do research and how to come up with research topics and techniques. I have a couple of different disciplines, some of which involve me being in a lab, dressed up in a lab coat like you see here, in kind of the traditional chemist's garb. We're doing our work, we're mixing gases, whatever we need to be doing that day. Whereas other roles that I take, I have to do my chemistry out in the field, underground, in tight spaces, and trying to learn about the different areas that exist and the different types of life that may exist in tight, cramped areas where we may not think of normal life existing. So, one of my roles, I said, was I'm an astrobiologist. So we can start off with, what does an astrobiologist do? That's a very common question I get asked. What do astrobiologists do? 30, 40 years ago, the answer was, I'm looking for aliens. And so you might have all these types of images that just popped into your mind. Not necessarily the accurate picture anymore. Now what astrobiologists do is we look for ways to look for life on other planets. We're trying to determine what is the best set of circumstances to determine life, existence possibilities on other planets. So we've got Mars. We're always sending new probes to Mars to take data. We have planets that we postulate what they might look like based on satellite data. And we think about whether they're Earth-like or not. And we've got multiple tools for doing this. We have Earth-based satellites. We have planetary-based satellites. So this would be a picture of Cassini, which went in orbit around Saturn for over 12 years. We have rovers that we send to other planets. We have deep galaxy telescopes that let us look out into the deep Milky Way and try to determine more information. And this is an even deeper one. So definitions. It's always good to kind of get the definitions started with, where are we going, what are we doing? So here's kind of a pie chart or a Venn diagram between astronomy, biology, and chemistry. So your classic subjects you might think about, looking at the stars, looking at life, looking at chemical reactions. So the astro kind of falls in between. We've got astrobiology, looking at the origin and distribution of life in the universe. Astrochemistry, looking at the reactions of chemicals and molecules in the universe. And then you've got where I fall in, astrobiochemistry, which is right in the middle of that little diagram, right here where everything kind of overlaps. We're studying the origin and the reactions and the distribution of the chemical elements and molecules important for life as we know it and life as we don't know it. So we're not looking for a life directly. We're looking for where are the molecules important for life being found and formed within the universe. So life as we know it versus life as we don't know it. Life as we know it requires several things. We need an energy source. And we need a source, which can be either solar, meaning direct photosynthesis, or indirect, we eat the plants, and geothermal. Those are two very common types of energy sources, some sort of heat that starts the life processes moving. Life on Earth is also carbon-based, comprising of nucleic acids that make up DNA and amino acids, which make up all of the different proteins in your body. And we know life on Earth is very water dependent. Life as we don't know it, we do postulate that there must be some sort of energy source that's going to drive life from one state to another. But that's about all we really can postulate about life as we don't know it, because we've never encountered life outside of Earth. So we can only guess what it might look like, what its nutrients might take. There are some things we think life can't do as a rule, but there's not a lot of rules aside from what we know about Earth life for life as we don't know it. So, let's jump to our solar system. Where did life arise? That's a good place to think about it, because if we can figure out where life arose on our solar system, maybe we can extrapolate that to other planets and other worlds. So, the earliest Earth, once upon a time, had no life, and it was very hazy. The atmosphere consisted of nitrogen, methane, hydrogen sulfur sulfide, carbon dioxide, everything that's coming out of a volcano. And these would form photochemical mixtures that would then lead to smog, soot, a heavy particulate atmosphere that you really don't want to breathe in. But those soot particles are very carbon rich. And carbon's one of the things of life. Now, if we look in our modern day solar system, we've got two other examples of these aerosol heavy atmospheres that are very hazy. We have Titan and Pluto. Each of these atmospheres are high in nitrogen, significant methane, and then a few other trace species that kind of add a little mix and variety. And both Titan and Pluto have aerosols forming that then fall to the surface. So the early Earth then, Titan and Pluto now, all had very similar ingredients. Now Earth gave life to life, but early Earth was much hotter than Titan and Pluto are. Early Earth, I mean, think about it. Earth is nice and comfortable. Titan is cold. Titan is cold enough that methane freezes. It's 96 Kelvin on the surface, which is very, very cold. Whereas Earth averages around 273 is the freezing point of water. A hot day might be around 300 or so Kelvin. It's pleasant to live. Pluto is even colder than Titan. So the coldness of an atmosphere will also stop the chemistry from moving as far forward. So it could be that the only thing lacking between Titan and Pluto versus the early Earth for creating life is just the temperature. Raise the temperature on Titan, maybe it's got life. Or maybe it's got a cryogenic type of life living on it. These are the types of ideas that planetary scientists work on. They try to compare the planets of now versus the planets of the past and try to draw conclusions about what that can possibly mean. It's called comparative climatology. And so a lot of my research has consisted of generating the atmospheres we know are around Titan and Pluto, and then extrapolating back to what that might mean for the early Earth. Especially because the early Earth's atmosphere is not something that is exactly known. So how does this work? So we take our initial mixture of gas and we give it either energetic particles or sunlight, or in our case we have lamps and electric discharges. We shock and irradiate them, drive that energy in, let those smaller molecules, nitrogen and methane, combine to form larger molecules, which then combine to form larger molecules, and eventually we get those aerosol products out, which then settle to the surface. This was originally started as a study in the 1950s at the University of Chicago with Urey and Miller, where they really wanted to understand how these early atmospheres could give rise to the molecules important for life. And so they took a setup similar to this, where we've got our gas mixture here, and we have our ocean stand-in here. They would heat the ocean, they would add electricity to their gas chamber, and then they would circulate. They would circulate the photosynthesized and statically generated products through the water, try to form some type of water-based organic that's active, and then they would pull off samples of this. And when they pulled off these samples, they were able to find multiple amino acids and multiple nucleic acids. So they found a lot of different pre-biological molecules that were being formed without the need for life to drive them forward, completely made by using electricity and water. Now, Titan also has a very interesting look to it. We think about weather here on Earth, and it's driven by the water cycle. Water goes from the ocean to the atmosphere. It makes the clouds rain, it washes the sediment into the ocean, it re-evaporates, and so forth, the big water cycle. Titan looks very, very similar to that, except because of its cryogenic nature, all of the ice, all of the water on Titan is mostly locked up as ice. But what drives the atmospheric chemistry of Titan is the methane cycle. We've got methane lakes and methane rivers. They evaporate into the atmosphere where they form clouds which can rain methane, or they can form these aerosols when we get this banding, when we get all this photochemistry going on, and they settle out as soot. So the water cycle on Earth is very closely mirrored by the methane cycle on Titan. So this is another reason why we're really interested in looking at Titan as a potential early Earth analog. It's got the same weathering processes, just a few different changes in the chemistry. So this is our setup. This is a setup of what an early Earth or a Titan-based chamber could look like. And you can see here on one chamber we've got it completely exposed to the elements, so it's whatever the temperature is of the lab. And that's our early Earth chamber. It runs at room temperature, and it does its work. The other chamber, the Titan chamber, is cryogenically cooled. We've got supercooled silicon oil that's bathing the entire chamber through these copper tubes, and that lets us drop the temperature down and perform all of these temperature exactly the same experiment between the two chambers. It's just one's at Earth temperature and one is at Titan temperature. And we get aerosols out. We get these clunky brown or gray or yellow based aerosols that come out of this photochemistry setup. And this is what was suspended in the early Earth's atmosphere. And this is something that then we try to tease apart what is actually in these molecules. What is in this gigantic goo that could potentially be giving forth life properties. And so we've been working on many different setups. Here in this GC-MS-MS, we've been pulling out different amino acids. We've been able to actually determine that there are pre-amino acids and some amino acids that are within the mixture. Not only that, but we can study how the formation changes with respect to temperature. So here is Earth normal, here is cooling down towards Titan-based temperature, and we get a very different formation rate. Here we get a very fast formation, here we have a very slow formation. So it's a much, much slower based reaction system, but we're still getting the same products. And we've been feeding these molecules to bacteria here on Earth, seeing, okay, we know we're making a whole bunch of organic goo. Is that goo considered foodstuff for life today? So could those aerosols potentially keep bacteria in the soil alive? So could that earliest Earth goo have become food for the earliest of Earth life? So kind of switching gears here. So that's a lot of the how we simulate chambers and how we use the chambers to look at comparative climatology between different planets that we can't visit. The other thing planetary scientists do is they go to the analog sites here on Earth and they study those sites, and then they compare what they learn at those sites to what environments might be out there in the solar system. So here we're studying wind cave. This is one of the big areas of study within my own research, is wind cave in South Dakota. So here again we've got this atmospheric cycling that's been going on. So that's a good story for how the atmosphere works on a given moon. But if we really want to understand what could be going on underneath the surface, we want to look at some of the other moons. What about moons where water actually is present in a liquid form? Now that sounds a little weird talking about moons with liquid water in our solar system, but we actually do have some. We have Europa and Enceladus. Both of these are ice moons, but they're by their gas giants, Jupiter and Saturn. Their gas giants stretch and pull them tidally, causing friction and heat, which then melts the subsurface ocean here. It also creates cracks and crevices where that subsurface ocean works its way up, forming undercrustal lakes. These lakes are then heated from below and irradiated from above, and they're full of organic-based compounds, trace organic, but they've got organic molecules that are important for life. So then the thought is, well, could we be talking about a system where the heat and the light come in and create the chemicals important for life? Much in the same way that an open atmosphere could create the important molecules for life, ice is just a window, still allowing several different types of wavelengths through. Could we be getting similar type organic chemistry in the water-based lakes? So here we can see a little bit more detail here. We've got the interstitial lakes here that are being formed, the ocean here, and then it's just working its way up through the cracks as the tidal forces pull and push the moon apart. The water ebbs and flows through these lakes. So just like on Earth, we have the rock cycle, where we've got continental crust drifting around and sublimating and spreading out to make rift valleys. We get the same story on these icy moons, and those are generating underwater currents, which are then creating these under ice lakes. So here in Wind Cave, we have very, very pure lake water, or at least the ability to find very, very pure lake water. And that means it's not generally accessible to terrestrial contamination. The lakes are about 500 feet below the surface of the crust, and life doesn't work its way down there by accident. You're not going to get a bat flying all the way down to the lakes. It's several kilometers just to get down there in the dark. So life doesn't find its way in there by accident, and it has to be brought in there. So that actually means that the lakes are very, very pure in terms of their chemistry and in terms of what life may or may not be existing there. So Wind Cave is a great place to study this because we actually have access to that deep underground aquifer with several of these underground lakes here. Now, the lakes need some sort of nutrients. We need to have some sort of input. Now, the lakes are cold, so it's not geothermal. So this is a very indirect solar pathway. Sun shines on the plants grow, and the plants die. When the plants die, their roots decay, and their roots are then, or the molecules from their roots, are washed into the soil. As it goes deeper and deeper, it eventually reaches the cavern system. And once it's in the cavern, it works its way down into the water table. And then in these couple of lakes down here, this WTHL and WCL, these are two different lakes. Those are accessible by humans if we climb and crawl our way down, and we can sample that water and see what nutrients are there and what may or may not be eating those nutrients. Those nutrients take the form of these very complex molecules, humic and fulvic acid. It's this brownish tan color. Sometimes you'll hear of somebody talking about tannins in their water. That's what this is. It's some sort of large biopolymer, and there's some very specialized bacteria that can break it down. Otherwise, it just kind of sits there until it falls apart naturally. So Wind Cave, let's talk about the cave. So Wind Cave is what's called a barometric cave. There are 150 miles of mapped caverns so far, and it's still being expanded. We haven't reached the end of Wind Cave yet. Now, my team is not the explorers who are mapping the cave. We go into the mapped sections and we do science there, but we're not actually the ones pushing new boundaries. We're trying to follow the survey team and then explore the life that is in the cave that they have then found. So barometric cavern, we'll kind of get to that here. This is what it means, that the cave pressure, all of that volume of the cave, all 150 miles of cave, are trying to go through one little hole every time the weather system changes. So the wind blows in, the wind blows out, and that's just the entire cave trying to reach equilibrium pressure with the outside. So it's a breathing cave. It can get as fast as 35 miles per hour going through that little teeny tiny opening. So here's an example of a very cold day on the outside, and the cave is breathing out, and it's breathing out nice, warm, and balmy air that is then warming the rocks around it. This often encourages creatures to come and crawl up in the natural entrance because it's 55 degrees in the entrance, right up until the wind changes, and then it gets really, really cold in the entrance as it's sucking in different temperature air. So we've taken 3 trips so far, and I am currently right now on my 4th trip, taking a variety of students to actually understand different aspects of the cave. So you can kind of see the teams here. We've been here off and on many, many times. So today, this week, would be the 2021 trip number 2, and we've got 8 students and 3 faculty working on this trip as it's going on right now. So what are we doing? Where are we going? We spend a lot of time in the natural entrance area. So this is the area where humans have brought in a lot of contamination. It happens. Humans are dirty. We don't wash our shoes. We're traching bacteria. Humans like to touch pretty things. So there's a lot of human contamination. So if we're going to understand how the bottom of the cave looks, we've got to understand how clean it is compared to the top of the cave. So we've been doing a lot of work understanding the top area of the cave to kind of start with. Where have humans touched? Did any of the human bacteria find a way to live there? Once we've kind of established that, then we can go down towards the bottom of the cave to try to understand the chemistry at the bottom of the cave. And so kind of to show you how we get to the bottom of the cave, it's a very long trek. We're going from this elevator shaft around this bend through some caverns and openings and down and down and down and down and down and down until we reach this lake down here. Now on this current trip we're going to push further. We're actually going to go over this first lake and continue through the passageway to this deeper lake here, Calcite Lake. So just to this first lake and back is a 4-mile round trip. To make it to the second lake adds almost another mile round trip total. So this is a very deep underground to try to get to. And it's not that it's 4 miles down, it's that it's 4 miles of twisting passageways. So how do we get to the cave? We have to prep, we have to practice, and we have to make sure that we can fit. So you build up your upper body strength, you build up your core body strength, you make sure that you've got your breathing control put together, but then you've also got to make sure that you can fit through the smallest possible space you can fit. We don't want to hit an 8-inch gap and find that you're 9 inches wide, because you're not going through that gap. So we've got a squeeze box to kind of give a simulation of what it's like to really feel that tightness on your chest. So here you can see that's an 8-inch squeeze, and that's what it takes to get down to the lakes, is to go through those 8-inch passageways. Very, very tiny, tiny, tiny areas to try to get through. Not a lot of people can handle both the mentality and the size limit of getting to that area. The gear we have, we need our helmet, we have knee pads and elbow pads, gloves, shoes with good grip, and then all of our equipment has to fit in these satchel bags that are under our armpits. So not a lot of big bulky equipment. We can only take small amounts of equipment, and any sample vials we take is taking up room that our equipment can't use. So it's a balance between what we can want to bring back with us for samples versus what we want to do there on site. We really try to do as much on site as possible so as not to disturb the cave and damage the rock features. But the water we can bring up with us to try to do more advanced techniques with. One of our key tools that we've been using a lot lately are these Lego-based measurement systems. It's using a Lego Mindstorm's brain, and we can hook that up to a variety of different probes to test all of the different properties of the water. And as you know, Lego is a perfect building block, so we can actually build and assemble all of these different probes right down there at the cave bottom. And it doesn't take very long. So we can actually have a 6-in-1 based instrument in order to take all kinds of measurements while we're right there at Lakeside. We have a variety of other probes. This kind of shows what it looks like when it's unpacked. We've got other flashlights, microscopes, Geiger counters for radiation, and all of this fits in that little satchel bag. We also have different kits. We have small UV vis spectroscopy kits so that we can measure how light interacts with the material of what we're dealing with. And we also have an X-ray based gun, so think phaser or tricorder from Star Trek. A little bit bigger than that, but it's what we have nowadays. And we point that into the rocks and minerals, and it tells us about the different content of those rocks and minerals, so we can understand how those rocks were formed or what layers may have been deposited on those rocks over time. So working in the cave is small, it's tight, you're going to bump your head a lot, so you can kind of see here some big wide open spaces, some very small spaces. So it's really kind of going to depend on how well you're feeling, how well you can keep your head together. There's a lot of slippery slopes, a lot of headbangers. It's always 53 degrees, wet, slippery, low clearance, tight quarters, and it's dim. We're doing most everything by our lantern light once we get off the path that was put in for the tours. And so we're doing everything by the light of our gadgets. So we want to make sure we've got good headlamps and we've got extra headlamps if possible if our batteries run out. So what do we see when we're in the cave itself? We see speleothems. So one of them, a big one, is Frostwork and cave popcorn, these beautiful aragonite crystals. They're sharp. They will puncture your skin like nothing else, but they're also very delicate. We see vugs, which can be full of these dogtooth calcite-based crystals. We also see flowstone that has been laid down by the constant dripping of water. In many caves, you'd be seeing these as stalactites and stalagmites. Wind Cave doesn't have a lot of stalactites and stalagmites because of how it's assembled. But we do see areas where we have the flowstone, which is the same composition, it's just flowed along the rock instead of dripping from the ceiling. Wind Cave, the big thing in Wind Cave, and this is why we don't see the stalactites, is we've got the boxwork. Boxwork is everywhere. And it's these veins of calcite that have been left behind when the limestone inside was dissolved by some process. And it looks like a corrugated cardboard box from the side, part of why it has its name. And you just see it everywhere in the cave. It's very uncommon though. Wind Cave has the most in the world. Jewel Cave nearby has some. Cody Cave has a little bit. But we really don't know a lot about how it formed. All we know is it's got a slightly different composition compared to the surrounding limestone. And there was some process that happened in the cave thousands and thousands of years ago that caused one limestone to dissolve while the other one stayed there. Now, for me, one of my favorite places to visit, and you can see it behind me here in my background, is Zebra Rock. It's this beautiful layering of rock. And here you can kind of see the size distribution. It's this huge feature, but it's way off trail. You only get to go there if you're on a scientific team or if you're one of the park ranger staff. But we've got these nice, beautiful bandings, and you can see these tans and browns and whites. But what's really perplexing about zebra rock, this is itself is perplexing, but what's really cool is when you turn on a black light, you see a whole other set of bands. You see yellow bands that go with the original bands, but then we've got these purple and blue bands that are going 90 degrees, indicating that there's some other layer of rock that is encasing the first layer of rock. As a matter of fact, when we turn on the black light, we see this sort of stuff everywhere we look. We see these fluorescents. So this is all done with black light flashlights. And we can just see everywhere we're looking, we see purples and blues and greens lighting up like glow-in-the-dark base things. Now the specific color that we see will tell us about what is actually causing that fluorescence. A soft green is more organic-based nature. It's those trace compounds that we are looking for that could be giving source to life. Pinks and reds are more inorganic, meaning metals. While those are interesting in themselves, they're not necessarily telling us about life-containing molecules, but more trace-containing other things. But you can just see entire walls fluoresce multiple different colors. So here's more examples of that trace organic fluorescence in the Flowstone. Anywhere you see Flowstone, we can find those organic molecules trapped in the rock when we use our UV spectroscopy. Here's an example of some of that red organic matter we were talking about, that red rock that has been encased in a rock face. And of course, green is one of the favorite fluorescent ones. These bright, poisonous colored type greens are always great to try to find in a cave using black lights. Now once we go deeper into the cave, because remember we're trying to get to the lakes, we see helicitite, which are these weird formations that are kind of growing out. They look like a coral reef structure. And it's thought that they originally formed when the cavern was flooded with water and some sort of bacteria formed these viney case things that were encased in calcite. And then when the water drained out, the bacteria died, leaving behind these almost coral-based features. And of course, then you've got the lake itself, which has this thin layer of calcite floating on top of it, the Calcite Rafts. So to actually get down to the lakes, what is it like? Well, you get a variety of different environments. Now, I showed you the squeeze box training, but that's only for very small portions of the climb. We're not actually spending all 4 miles crawling through very, very tiny openings. We get these beautiful large galleries that we get to go through. And that's one of the best parts about being a cave explorer. If you can fit through the small gap, you might be lucky enough to discover a brand new chamber on the other side. So to actually get to the lakes down at the bottom, it's a 6-kilometer round trip, or about 4 miles. It takes us 2 hours to get down there, and we have to go from the natural entrance at the surface all the way down to the lakes and then back up again. So the first half of the climb is more along what I've already shown you. We've got the box work, we've got the frost work, and eventually we get to Buffalo Gap, which is the halfway point. It's pretty straightforward from there. But then once you get to Buffalo Gap, that's where the technical crawling becomes. We have to go through the boxwork chimney. And the boxwork chimney is this vertical gash in the cave that's very narrow. As you can see, my student here is pinched between the rocks. Here you can see the cave guide, my student, and my feet here. And you have to go down this 75-foot chimney of cave formation in order to get down to the lake level. So kind of give you an idea of what that looks like from the bird's eye perspective. You can kind of see it's not for the faint of heart. If you are claustrophobic, you do not want to try to go to the boxwork chimney. This is one of the reasons why we do all of that squeeze box training. We need to know that when we hit this chimney and the cave guide says this is 10 inches of clearance at its worst, then I can say, well, I can do an 8-inch gap, so I can fit So it's a very mental relief knowing I can fit through here if I get my body twisted the right direction. So at least that load of I'm not going to get stuck has been lifted. So a lot of it is just finding the right handholds, finding ways to kind of slide our way down. You can see here I got to slide sideways a little bit to try to get through a slightly wider opening. And then we work our way down and down into slightly larger areas. So this is still just that boxwork chimney. All total, it takes about 12 minutes to get just down the 75-foot shaft. Now eventually when you're down at the bottom, you come through a few last tiny little holes, and you come out into the big chamber at the bottom of the cave, and then you follow these passageways to the lake. So this is one of the really big, exciting things about the cave exploring is you finally get out of that narrow passage and it just opens up into the large vault. So now once we're walking around in the lower cave, you can see it's a much more open type system. We've got large walls. We do have to climb over a few of the larger edifices to get around. So this one is called Garden Gate. We do actually have to climb up and over it with a wall climb. We've got our rope in place. We haul ourselves up to the top of the cave, throw the rope over, and then climb ourselves back down on the other side. And it's this nice 30-foot climb at the bottom of a 400-foot deep cave. And even here, you can see the gear bags are strapped onto everybody. Every one of those gear bags have some sort of sampling equipment, some sort of scientific equipment, or our sample vials so that we can bring the water back up to the surface. It also has all of our food and water that we as a team need, because we're down there the entire day. So we need our lunch and we need water so we don't get dehydrated while we're down there. So we have to fit a lot in these little cave bags, and everybody has one, generally speaking. So then we're at the lake and we're able to do our sciences, we're able to sample the water, and we're able to get an understanding of what's actually going on within that water system itself. And of course, we always stop for the fun photo ops while we're down there, because it really is an accomplishment to try to get to some of these places. This year, the trip is not just stopping here at this rock peninsula on this lake. We're actually going to then go up into this opening above the lake and go to the other side, and that's the passageway that takes us into the next lake, Calcite Lake. So the water sampling we did, we actually took a lot of samples from the stream, as well as sampling the water in the lake itself. And we do plan to go further. There is a large area of dry cavern in between the WTH Lake and Calcite Lake further down. So we've got another half mile of climbing yet to do in order to get to Calcite Lake. So they're completely separated by cave, but they both tap into the same water source. And so one of the questions is there life in the lakes? Which the answer is yes. But then how close are those two life forms in the different lakes comparing? Are they similar in nature, or are they not similar in nature? We know there's life, we know there's nutrients, but is it the same life between the lakes, or is it different life between the lakes? So that's one of the questions we're trying to answer with this trip. Remember, this is extreme life. So we know that life can live down here. That means life can live in a similar environment in the solar system. So what have we found? Well, we found organics everywhere. Remember, organics is carbon-based material. Carbon-based material is what is important for life. That's the DNA. That is your proteins. All of that is in those organics. And we find it everywhere in the cave, and it lights up beautifully under black light. And we can get all of this beautiful data. It's in the water. Everywhere you look, it's in the water. And this is just the spectroscopy data showing that that beautiful blue color coming off of the water actually has a wavelength associated with it for what is the length of the wave of light coming off of it. And it's a very unique wavelength to that shift. And we can play around with concentrations and actually go from super concentrated at the surface down to very weak down at the lakeside. And that actually lets us think about how many nutrients actually make it all the way down to the bottom of the cave from the surface. It's not a lot, but it is just enough to support life. And that's what's important. There's just enough nutrients that life finds a way to live down there. So again, showing some of the organics at the bottom of the cave, more organics under different colors. Even the calcite rafts themselves are concentrating the organics from the water, which then react under black light. So we're actually forming all kinds of really interesting organic locks and concentrations down in the cave. Now, the other thing we've been doing is we've been trying to figure out how to recreate those interesting colors in the cave. So we've got, these are the data from the cave. This is all of the different colors of light coming out of the different colors of rock. Like behind me, we can see the purples and the blues and the greens and the yellows. This is what it looks like when you start looking at the different colors on a scientific scale in terms of the colors of light coming off of something. And we can recreate these things in the lab. We can actually go into our lab and we can force water to create these crystals. It's just calcium carbonate, which is the hard water gunk that is around all of your faucets at home. So we actually have a way to make that and force it to happen spontaneously. And in doing so, we can actually trap other species in it. We can trap organics and we can trap inorganics. And so you can see when we trap the inorganics, we can get these brilliant purples and pinks. When we trap the organics, which even includes coffee as an organic, we get these greens and these blues, which are very similar to the colors we see in the cave. So all of these are synthetically made crystals. We make them in the lab, and we can see how beautifully they match the colors that we see at the cave, which is fascinating. At Zebra Rock, we saw all of those different colors, and here we can kind of see what those would look like on the spectrometer basis in terms of the wavelengths of the different light. We've got our blues and our greens, and we've got our reds and our pinks down here, and that's what it looks like. But then we can recreate those colors, the pinks especially, we've been working on. We can recreate those pinks almost exactly by doping in a little bit of manganese. And we get these beautiful pinks. So now we're talking about going from the cave, looking at that data, moving back to the lab, where we're going to recreate it using synthetic analogs so we can better understand it without having damaged the cave. We took the data in the cave and we left the cave alone so generations can enjoy it. Then we come back to the lab and we bake our own little cave crystals and we analyze them and see if we got the same match. Once we understand what made the cave crystals, then we can start saying, okay, so if I saw this color on another planet, what would that mean? Could that be a potential indicator of a life sign? Here's that brilliant green we talked about. Now this brilliant green I sometimes like to call Disney poison green. So here we can see 3 different spectra all lined up with the same shape. We've got Garden of Eden and Fairgrounds. Those are both in the cave. And then we've got this geode, which I have in my private rock collection. This geode, that brilliant green, is from trace uranium. And I've tested this geode several different ways to confirm the presence of uranium. And it gives this shape of spectra with all of its green. So we now know that on those 2 ledges in Wind Cave, the Fairgrounds and the Garden of Eden, there is some trace uranium causing this brilliant green fluorescence. Now it's so trace we can't even get a Geiger counter to register. So nobody is in danger of getting any type of radiation damage from it. But it's fascinating to see how this beautiful shelf in Wind Cave, which is beautiful in natural white light because it's all these white fine crystals, flip on the black light and you've got brilliant green. Then we go home, we don't even have to take a piece of the cave with us, and we look for any source of bright green fluorescence in a mineral collection, and we can match it to a uranium-based mineral exactly to a T. And of course, the cave is just full of microbial life. We've got the prairie surface, we've got the lakes, we've got some muddy and sediments down there. Microbial life is everywhere within the cave, from top to bottom, from easy to track in by accident by humans and animals, down to the very, very depths of the cave where it has to have been living there on its own for thousands and thousands of years off of these trace nutrients that make it through the surface. So what does this mean? That means that if the right situations arise on Europa and Enceladus, we could be looking at a situation where all of the constituents really are just right for the chemical formation of life-containing molecules. We could be looking at amino acids and nucleic acids forming in the oceans of Europa or Enceladus. And if the pre-biological molecules are there, who's to say what can't happen there? Somehow here on Earth, the pre-biological molecules became biological life. Somehow. We don't know that step. We may never know that step, but that's one of the big questions. But what we do know is there's a very good chance that the same pre-biological molecules are present in other situations. So there's always more work to do. This work is funded through 2024. So we've got several more years of funding in process. And so if you want to know more, the University of Northern Iowa does have a museum exhibit about this. You are more than welcome to come out and visit. Or I will leave my contact information with your organizers, and you are more than welcome to contact me there with any additional questions about this research project. So with that, I want to thank you for your time and for having me give a talk today. And I hope you continue to enjoy your exploration over the next couple of months.